Let's get started. I hope I am audible to everyone. Welcome everyone to the European Industry MedDRA Users Group webinar. I am your host today, Amit Ranjane, EU MedDRA UG, and we also have Destiny Hutcherson and Brian O'Hare from the MSSO team for the technical support. Some basic information about the questions. If you would like to submit the questions, pop out the control panel, which is located on the right upper side of your screen. Type the question in the question box and click send. Your questions will be addressed at the end of the topic and also during at the time of the Q&A session. Due to the time constraint, we may not be able to answer all the questions submitted. However, we'll try to make all the questions as possible to answer those. Common questions that we get around slides and recording. The slides and recording of this webinar will be available at the MedDRA website shortly after the webinar. The link is provided below. And before we start, we'd like to say a warm welcome from the User Group Steering Committee. A lot of known faces here for the people who attend the meetings and webinars on the regular basis. For the people who are attending for the first time, on the behalf of the team, we would like to say hello to everyone. And before we begin, we would like to ask some polling questions. These questions help us to decide the type of webinar that we would like to have in the future. So I'll request you all to take the polling questions first. The first question that you will be able to see on your screen is, from which region are you attending this webinar? You will have approximately 30 seconds to answer the question. And Destiny, let us know the results of the first polling question.
Okay. Okay, so we have 68% from Europe, 15% from North America, and 9% from Latin America and the Caribbean.
Thank you, Destiny. We move on to the next polling question. What type of organization do you work for? And the polling starts now.
Okay, we have 52% pharmaceutical development manufacturer or distributor, 26% CRO service provider, 13% other, 8% regulatory authority, and 1% medical device development organization.
That's a good representation from the pharmaceutical industry, I would say. We move on to the last question, polling question. What is your role or position in the organization?
Okay, we have 69% in drug safety pharmacovigilance. We have 14% in coding, 8% statistics programming, informatics, or data management. We have 4% in other, and then 4% in clinical research and operations.
Perfect, Destiny. Thank you. We see great attendance there, diversity in the attendance. Okay, so then we can move on to the agenda topic for the day. The topic for today is utilizing semantic similarity for pharmacovigilance by Jeffrey Painter. A brief introduction about Jeffrey Painter. Jeffrey is the Senior Director, Safety Innovations and Analytics, and the quantitative leader for global safety at GSK. He has 18 plus years of experience in drug safety and statistical genetics research and development, with a background of software development, mathematics, and statistics. He holds a bachelor in computer science and applied maths from North Carolina State University, Juris Doctor in North Carolina Central University School of Law, and also an MS in computer science from the Georgia Institute of Technology. With this introduction, I'd like to hand over to Jeffrey for the further presentation. Thank you. Jeffrey, over to you.
Thank you, Amit. Before we get started, if you'll bear with me, I'd just like to give a little bit more about my background and how we got to where we are today. This has really been a long journey across my career, and I started at GSK back in 2006 as an intern in a new group called the Semantic Technologies Group. I've been fortunate to have some great mentors over the years. The first was Dr. Gary Merrill, who holds both a PhD in philosophy and computer science, and really sparked in me my interest in ontologies, medical terminologies, and really leveraging my computer skills and programming to make an effort in improving the lives and safety of patients everywhere. Fast forward a little over my career, I worked with Dr. Matt Nelson, who at the time was the head of statistical genetics at GSK. He had learned of the work that Dr. Gary and I had done with biomedical terminologies and said, hey, we have similar problems in genetics and drug discovery. And so I had the opportunity to work with them. And that's where a lot of the foundations of some of the semantic similarity I'll be sharing today started. Then around 2011 to 12, we saw an uptake in the use of social media, and we became really interested in, from a drug safety point of view, how we could, investigate these massive amounts of social media posts, potentially looking for early signal detection methodologies. And that's where I really started to develop my skills around natural language processing and looking at how we might take the way that people talk in everyday language and map that to standard terminologies like MedDRA. I left GSK in 2016. I joined a startup in Boston. I spent 2 years working on advanced AI and ML methods for modeling Parkinson's disease and disease phenotyping, working along with our wet lab biologists and trying to connect the data we saw in the lab to observational data, again, leveraging a lot of the skills in ontology and terminology work. And for the past 4 years, I've been fortunate to come back to GSK. I met what I would say is my third mentor of my career stage at this point, Dr. Andrew Bate. And Dr. Bate leads the Safety Innovation and Analytics Group. We're a very small team. We have 2 expert statisticians based in Belgium, Francois Haugenet and Olivia Mahot. We have a clinical expert, Dr. Greg Powell, and we have a real-world evidence expert with MedDRA. And finally, we have a regulatory expert on the team, Vijay Kara. And I serve as the quantitative leader. And a lot of what we're focused on has been, I would say 4 years ago, focused heavily on AI methodologies. If you'd asked me 20 years ago what AI is versus what we all think of it today, it's drastically different. I will say what I'm presenting today is not really AI in today's terms, but 20 years ago, it might have been considered AI. And I think the only thing that will not change is in 10 years, we'll have a totally different definition of what AI actually means. So with that, I'll get into the slide presentation. I've tried to keep this at a level for a general audience. Of course, if you have questions, please note them and we'll try to address them at the end. So the topic today is how we're utilizing semantic similarity in drug safety or pharmacovigilance. And we're doing this, oh, let me stop sharing the camera. Okay. And we're not doing this in a vacuum. We do have a very specific use case. We were looking at how we might improve our ability to do signal detection. So signal detection is the quantitative evaluation of all the safety reports that we get and that we're constantly evaluating in post-marketing situations to understand what the safety profile of our medicines look like. And traditionally, the state of the art in signal detection up until today has been what we call disproportionality analysis. So looking at odds ratios and essentially seeing, if we're looking across spontaneous reports of safety events, is it the case that a drug adverse event pair is likely to be explained by the medicine or not? And so we look at the frequency of occurrence of those reports. But the problem with that is that traditionally, that analysis has been done in a strict hierarchical fashion, typically with the MedDRA terminology. And so you may, filter down by the SOC, HLGT, or HLG classifications of the MedDRA PT terms, but it's really a binary yes or no inclusion. And our goal was to see, is there a better way that we can understand the relationship between the safety reports as they're the safety events as they're reported versus what the actual signal might be in the noise. With that, also in my background, I did minor in statistics and cognitive science. I had a very strong interest early on in how language is represented and how the mind represents knowledge. And so that gets us into the definition of what is a semantic network. So at a high level, the semantic network is a knowledge structure that depicts how concepts might be related to one another and how those concepts interconnect. So semantic networks are useful today in AI for data mining, connecting various concepts and calling attention to different relationships among medical concepts or what have you. In the early days, these networks were quite simplistic. They were used to create categories of known word relationships, typically as synonyms and hyponyms. The definition of what is a hyponym, it's really, that broader or narrow word relationship between terms, where hyponym is just a word that has a more specific meaning than a related general term. The typical example here is bird is a hyponym of the word animal. And so this idea originated with Collins and Quillians, who were cognitive researchers in the 1960s. They wanted to try to build a model organizing memory structures in the brain. And they did this, quite rudimentary with pen and paper. They proposed these networks of concepts where the links represent different relationships, things like is a, part of, and simple associative connections. So for example, a bird can fly, a bird has wings, a fish has gills, and a fish can swim. And so we start to see how they're breaking down the mental relationships between objects and their properties in these semantic network models. These advanced in the 1970s when researchers in AI started to look at these concepts and adopt them in what they called their early natural language systems and reasoning engines. This is when they first attempted to automate inferencing using conceptual hierarchies. And by the 1980s, there were extensions to these networks called frames and scripts, which were also developed by cognitive psychologists for helping us to understand how human language understanding and development occurs over time. And the goal here was, could we teach computers how to think and reason over language the same way that humans do? Then by the late 1990s and early 2000s, the internet was sort of blowing up. Tim Berners-Lee famously created the World Wide Web at the CERN laboratory. And as he was developing it, he said, you know, we really need a way to expose data and make it more accessible and discoverable on the internet. This was, you know, very pie in the sky thinking, and as a result, they started creating what are called the RDF and OWL languages. These are resource description frameworks, and OWL is the web ontology language. And really, these are just easier ways to make data accessible for computation. This is also the era where the UMLS became deeply important for healthcare because it brought together disparate medical vocabularies into a single interconnected semantic network. And so today, we're not reinventing semantic networks. They're highly mature. But we're finally able, with today's computational power, to use them at scale efficiently and start to apply them to domains like drug safety. If you're interested, there are some publicly available semantic networks. As I mentioned before, the UMLS is probably the one most important to our field. And the other being WordNet, which was developed by Princeton University. WordNet was really designed in the similar concept to the original Collins and Quillian research of trying to take all of human language and put it into a concept network graph. These were highly used, I'd say, in the early days of natural language processing in the 90s and early 2000s, and really predate what we see today with technologies like ChatGPT and these large language models. Whereas the UMLS really took a hands-on approach to collecting all of the known biomedical terminologies from SNOMED, ICD codes, MedDRA, and trying to put a universal concept network on top of those terminologies. And where I really got started in my career was, you know, I joined back in 2006. We were working with epidemiologists who were still relying, if you can believe it, on printed manuals of ICD codes and CPT codes. And they needed a better way to interrogate the data in observational databases. So at that time, we put together a system we called CodeSlinger, which there are some publications we had many years ago. But CodeSlinger essentially was a desktop tool that we put on the epidemiologist's computer where they could say, I'm interested in the concept of myocardial infarction. How would I find those terms across MedDRA, ICD, or SNOMED codes, depending on the database I'm investigating? And we really utilized the UMLS's concept network to help power that in the back end. And we found it a very useful tool to help basically bridge the gap between medical terminologies. So we can't talk about semantic networks without talking about distance. And what does that mean? So once we have this semantic network, we want to be able to compute distances between concepts. This helps us to understand how potentially related one concept is to another. Whereas in MedDRA, you have this hierarchical relationship going from the system organ class down to the preferred term. That terminology structure was really designed for quick and efficient coding of safety events, but not necessarily with the clinician's point of view of, I want to search for, you know, any related concept to myocardial infarction across my safety data. It's not necessarily the case that the MedDRA hierarchy is going to give them exactly what they're looking for. And so we look at semantic similarity and distance as a way to help us find the relatedness between any two concepts in this network. The introduction of sophisticated statistical and corpus-based approaches appeared in the late 90s and early 2000s. And I would say a lot of that research kind of went away around 2015, 2016. I think largely in part due to computational restrictions. It just became very cumbersome to compute these highly complicated statistical measures across large amounts of medical concepts. We have seen more recently advancements in trying to integrate machine learning and deep learning techniques to help us improve our semantic analysis. And I mentioned this paper here published in 2023 in JAMIA by Dr. Mal, and Olivia Bodenreiter, if you don't know, has recently retired from the National Institute of Health. But I had the pleasure of meeting Olivier many times at conferences over the years, and he was instrumental in essentially steering the UMLS across the past 15, 20 years and doing some great research into the semantic networks and representations in the UMLS. So in this paper, they looked at using AI to take what had traditionally been a very human-intensive manual process. So anytime you would add a new code to MedDRA, the UMLS team would ingest that information and then try to figure out where does it fit in relation to all the other concepts. And so in this paper, they're starting to look at how we can utilize AI and LLM technologies to automate that step and ease the burden. They showed some really promising results there, and I encourage you to check that paper out if you're interested. So when we talk about distance, I do have a math background. I'll try not to get too mathy here, but I've provided some formulae here on the right to explain essentially from a mathematical point of view, when we talk about distance, the simplest distance is Euclidean distance. So what's the distance from point A to point B if you were to simply draw a straight line? But there are other types of distance measures. We start with those that are called path-based, and that essentially means counting the paths along the graph. And so if you were looking at two preferred terms that shared the same parent high-level group term, they would have a distance of 2 because they just simply are siblings. But they're more statistically complex methods that we have developed since then that work based on what are called information content based or IC based metrics. And some of these, and you'll see a lot of names here, so Wu Palmer, Leacock and Chaudhary, Rada, all of these measures are essentially named after the inventors or the authors of these methodologies. And so whenever you're talking about distance metrics, if you refer to the Lin measure or the Resnick, those are the statisticians who originally created those methodologies. And so we really hadn't, to this point, investigated which of these types of measures works best in drug safety. And so a lot of the impetus for what we're going to get to next in our experiment that we recently presented at the AMIA was understanding if we were going to utilize these systematically across our drug safety data, which metrics should we really use? As I alluded to before, these have been used in the past. As I said, Dr. Matt Nelson was the former head of statistical genetics at GSK, and I worked with him on this paper published in Nature Genetics called The Support of Human Genetic Evidence for Approved Drug Indications. And he came to me and basically said, Jeff, we have all this observational data. We'd like to link it to known gene indications. And so for this project, we didn't use MedDRA, we used MESH, the medical subject heading terms, to try to identify trait similarity. And at the time, the only tools really available to us were this Perl programming language, which was very popular with biocomputational scientists in the late 90s, early 2000s. It's since fell out of favor. You might think of it as today's modern equivalent of the Python programming language. But Dr. Ted Peterson in the University of Minnesota created this package called UMLS Similarity, which essentially allowed you to set up your own copy of the UMLS on your computer or server. And through this programming interface, you could compute these distance metrics. And they had implemented a lot of the measures we talked about before, those path-based and intrinsic-based metrics such as Lin and Resnick. And we were able to utilize this package for this study to compute, and I think we only looked at about 5,000 clinical terms from the MESH terminology. And so when you start to compute these distance metrics and you want to create a matrix of all the distances between these 5,000 terms, that starts to grow exponentially large. And so it required over 74 million computations just for this 5,000 term set. And it literally took hundreds and hundreds of hours of compute time and was, very compute resource intensive. But ultimately, we were able to show there is value. We were able to demonstrate that through this similarity measure, there was strong evidence to show that drugs that targeted specific gene indications were highly more successful over the course of their drug development cycles than those that were not targeting known gene indications. Now, I will say, while this was not specifically looking at the different metrics per se, we relied on what was then known in the publication in the literature, which told us that Lin and Resnick were generally considered the best metrics at the time. We just took that on faith. It was, as I said, hundreds of hours to compute these computations, and we really didn't have the time or resource to evaluate every metric. But even with just the Lin and Resnick scores, we showed there was promise in this approach. Fast forward a little, in 2015, Dupuche and Graybar published a paper in the journal Biomedical Informatics, and they presented something that was very interesting. They took the MedDRA terminology, which is shown here on the left, and they took the SNOMED ontology and essentially created a conglomeration of the two, where they projected the MedDRA terminology onto SNOMED to try to enhance it. And they called this their onto-EIM structure. And they were really looking at, can we use these semantic networks to replicate standard MedDRA queries? They didn't really do a systematic analysis across the different metrics. I would say, unfortunately, their results were less than stellar, but it really paved the way for a new way of thinking about how to represent MedDRA in different terminology systems. And we used part of this in our experimentation. So fast forward, we know this old Perl code is excruciatingly slow. It was built over 28 years ago. There's been no real improvements since. And I was really trying and struggling to find what is an alternative process that we can do to create these semantic similarity metric computations much more efficiently. I started digging and I found the Apache CTAKES project. This is an open source project that was designed around clinical text and NLP processing. It's mainly maintained now by researchers at the Mayo Clinic. And buried deep within their code was a small project from Yale called YTech. And YTech essentially did what I was trying to accomplish. They created concept networks utilizing the UMLS. But the code was broken. It had not been updated in over 10 years. I spent several weeks digging through it and piecing it back together. And as of today, I'm happy to say we've now built on top of this platform our new semantic similarity calculator that allows us to compute across essentially all of MedDRA, 26,000 PTs, in minutes rather than hundreds of hours. It also allows for us to start to look at different representations of MedDRA, which I'll get into when we talk about the experiment that we ran. So I just got back from the AMIA conference where I presented our ontology-based semantic similarity measures for clustering medical concepts in drug safety. And I'm going to review these slides quickly with you. We've already talked about what a semantic network is, what these semantic similarity measures are, and how we had used them in the past for both genomics and other areas, but they have yet to be really utilized for pharmacovigilance and drug safety. We understand this MedDRA rigid hierarchy, and we're really trying to move beyond that using these more enhanced networks. And we see, for example, neutropenia versus bone marrow failure fall within totally different branches under the MedDRA hierarchy, even though neutropenia can be a sign or symptom of bone marrow failure. And we hope to show that by looking at these semantic similarity metrics, we could offer a scalable data-driven approach to identify clinically meaningful groupings. So how did we do this? We looked across various metrics. We also looked at different integrations of MedDRA with different terminologies. We integrated it with SNOMED, with MESH, with all three together. We looked at MedDRA alone and really did a systematic analysis to determine for our drug safety needs, which metrics would perform best. And we're going to use this later when we talk about how we're using it in our signal detection methodology today. So we tested 6 different metrics. We looked at 2 path-based, the LCH and WooPalmer. Again, remember, these are the names of the inventors of the metrics. And we looked at 4 IC-based metrics, the Resnick and Lin, which the literature had originally told us were some of the top performing. And we also looked at the LCH and a method called SOCAL. We wanted to understand how the different weights of similarity could share concepts within the hierarchies. And so we selected 12 narrowly defined standard measure queries where the SMQ name had a matching PT. So for example, if the narrow SMQ was for Parkinson's disease, we knew there was a PT that also had the name Parkinson's disease. And we called these our centroids, or the central concept of the SMQ. And we needed a central concept to compute the distance from, right? So you need to know where you're starting and where the distance metrics will fall from there. So in our minds, this made logical sense that, you know, we pulled these 12 narrow SMQs, we find their centroid PT, and then we created the pairwise similarity scores across all 26,000 MedDRA PTs to that centroid. This was not a light undertaking. We had 6 reviewers from the UNC Eichelman School of Medicine who were 3rd and 4th year pharmacy students trained in medical coding, who collectively evaluated over 6,000 PT groupings, taking around 220 man hours. And we finally then benchmarked this against the SMQ inclusion. And we also looked at if we were just to take a straight HLGT grouping, how would that compare? We've already talked about the difference between these path-based versus IC-based, but this is just another visual representation showing you the path-based are simply counting the hops between the nodes on the graph, where IC-based is taking a more information content-based approach, looking at the overall structure of the data, not just how many steps there are in between it. So I won't bore you with all the statistical details, but it's a much more complicated way of computing distance. So what were the results of this experiment? After we had all the data, we computed what are called F1 scores. And so in machine learning and statistical analysis, an F1 score is a measure of performance. A higher F1 score is better. And then you see also on this graph, the numbers from 0 to 1 are the SSM values. So we're trying to find what's the lowest distance that we would consider for including a term based on its semantic similarity, but also the highest performant in terms of what the experts told us should be included in that study. So let me just clarify, the task that the students were asked to do was they were told, here's the SMQ, here's the primary PT term. If you were doing a safety study on Parkinson's disease, what are all the terms that you would include in that study? And we presented to them through a web interface. They could select to include or exclude these terms. And it included all of the terms from the SMQ, as I said, the HLGT grouping that PT fell within, along with the semantic distance terms. And I think we limited it to maybe 600 or 700 terms based on distance measures that were selected at random for them to select from. And the really surprising results here were that both Lin and Resnick, we see here are the green and orange lines, really were not the top performers as the literature had told us in the past. We found from our experiment for drug safety in particular, the SOCAL metric was the highest performance, giving us an F1 score of 0.403, and then WooPalmer and LCH falling quickly behind. But ultimately what we see here are these IC-based approaches are more performant than the path-based. And when we looked across the different representations of MedDRA in combination with SNOMED or MESH, we started to see there are bimodal distributions suggesting that there are ontological structural effects depending on what representation you put MedDRA within. Overall, we felt this really improved our ability to do cluster analysis and better aligned with clinical meaning based on the students' analysis. Further, every code set was evaluated by 2 independent students, and if they disagreed, as I mentioned, Dr. Greg Powell on our team has over 30 years of drug safety experience. He was the final adjudicator if there were any discrepancies. And the data set that we generated from this is available on our GitHub. And so if you're interested in these results or replicating the analysis, all of that data is available. So we took from this experiment that we really had a better understanding, a systematic analysis across all of these different metrics, different representations of MedDRA. We ultimately determined, this will support analysis, but it's not going to replace expert curation. They're not perfect. But when you're doing high throughput screening and drug safety signal review, this is really going to reduce our workload and improve reproducibility. Potentially, we might look at other ensemble methods going forward, but all of this has been used to fuel what we're calling our new semantic similarity informed Bayesian borrowing for quantitative signal detection. The paper is available as a preprint. If you're interested, the link is there. And I'm happy to say we have submitted it to the Journal of Statistics and Medicine, and it's under review and hopefully should be published in the next year. So what is this Bayesian borrowing all about? We're calling this our novel quantitative signal detection method. As I mentioned, Francois on our team is our expert statistician. I don't claim to be the expert statistician. So my role in this has really been in support of this analysis, building the semantic networks, and building this next piece, which we're calling PV Lens, which we really needed a good drug reference database. The gold standard before this was called CIDR, the Side Effects Research Database, which was largely abandoned in about 2016 and had not been updated since. And so what we have done here is we took the FDA's SPL, that's the structured product label, and created an extraction pipeline. Again, there's no AI or machine learning involved in this. We're using traditional natural language processing, and we're essentially extracting from all the labels of medicines, the indication section, the adverse event section, and the black box warning sections. And the pipeline, what it does is it scans all of that text and it tries to align it to MedDRA terms. And so, we work in drug safety. MedDRA is our lingua franca. If we're dealing with FAERS data or the EU data, all the medical events are coded in MedDRA terms. So we really wanted to know for every medicine, what is its known labeled indication and adverse event profile. And what this also allows us to do is we've created a temporal timeline so we can tell you when a new adverse event was added to the label. And this was critical for us to do the next step in our safety signal evaluation experiment. We needed a large enough sample data set of products to know when a product came onto the market and when a new labeled event was added after it was on the market. I'm happy to report that this is our first open source project from the GSK Global Safety team. The entire pipeline code is available. If you're interested in building your own version of the PV Lens database, all the code and methods are there, which is in stark contrast to CIDR, which was never really validated or able to be replicated because the source code for that system was never made available. And so we're really hoping to extend this internationally. We have colleagues at the University of Copenhagen who are hopefully helping us to add the EU labels, and we're encouraging anyone who's interested to get involved on this project. So now I think it's time to show a nice visual representation of what do we mean when we're talking about these semantic networks. Again, I go back to this example of Parkinson's disease, which is near and dear to my heart. I spent 2 years working on PD, and really what we're showing here is a visual representation of what these semantic networks look like. So at the heart of it, we see the main PT code for Parkinson's disease. And each of the nodes represented in blue come from the narrow SMQ definition. And each of the triangles in yellow come from the broad SMQ definition for Parkinson's disease. And so we see here what the semantic relatedness of each of those terms looks like. And so for the central concept PD, we see bradykinesia and Perry syndrome are directly related. But we see far to the left, hypertonia and muscle rigidity are a couple hops away from Parkinson's disease. Now, if we use our capabilities of semantic similarity, we would like to see what does it look like if we extend this network by incorporating concepts that have a measure of 0.75 or greater, and we start to see these pink diamonds appear. So decreased activity and multiple system atrophy are concepts that are brought in via the semantic similarity that were not originally included in the SMQ definition. And so essentially what you're seeing here is the kind of data that these students were evaluating to understand what are those thresholds of similarity that we should establish if we're going to run this at scale. And I think the big takeaway here, again, I won't get into the deep statistical methodology of what Bayesian borrowing is, other than it is a way to do information borrowing using the semantic network. So rather than traditional methods where we're looking strictly at the rigid MedDRA hierarchy, we're using these semantic distances to pull in and weight information from various relationships between these concepts to help us evaluate the safety data. And so in this experiment, we looked specifically at data from FAIRS. We found through our semantic similarity analysis, we could identify roughly 70 products that were launched between 2015 and 2017. So we were looking pre-COVID, and we wanted to know of those products which had at least one or more new adverse events added to the label within 3 years after its launch. And so having that baseline data from our PV Lens database, we were able to test then this methodology compared to existing methodologies. And what we were able to show, which is pretty clear on the graph here, is that our ability in time to detect, so we want to know as soon as possible when there's likely to be a new signal from that drug adverse event association that would warrant an investigation from our safety team. When we use the semantic similarity in combination with Bayesian dynamic borrowing, we showed that consistently we could identify more true positive signals on average 5 months sooner than the traditional approach. And so this gives us great hope that this is a potential way forward in improving our abilities to identify potential safety signals earlier. And with that, I'm going to call it a wrap.
Okay, so thank you, Jeffrey. I think we're now in a position to take any questions that you might have. So if anyone has any questions about the presentation, please send them into the chat. I don't see anything yet, but if anyone has any questions, please send them in. I guess, Jeffrey, maybe I can ask a question of you.
Yes.
So this method that you have developed, is it my understanding that you're still trying to position this within the process of your safety analysis? In other words, are you starting to actively use this now, or are you still trying to hone what you're trying to do to get to the point where you feel it's a viable option for you as you analyze your data?
Yeah, I would say the proof of concept stage is shown that there's promise here. It's still not perfect. We know that, you know, it has shown promise in detecting signals earlier than traditional methods. I will say this is a much more computationally intensive process than typical disproportionality analysis. And so rolling this out is going to be probably a year-long effort to implement. Part of what we're working on as I speak is the semantic similarity calculator that I talked about. We're in the process of getting legal approval to open source that code. I think that might really open the door to help others who are interested in the space potentially collaborate with us, do more experimentation, look at their own data. You know, we've demonstrated, I think, fairly well that, you know, these similarity metrics that we did the evaluation with, we've established some strong thresholds and criteria for how to understand which ones are best used in this process. But that doesn't mean that, you know, it's the definitive answer. And so I would say at this point we are circulating the information with the community. We're talking with regulators. We're planning to do presentations with FDA and EMA to get them comfortable with this methodology and to get some buy-in that, you know, this is a potential new way that would be acceptable to the regulators going forward.
Okay, well, thank you for that. Well, we now have a bunch of questions, so let me start by reading the first question out, which is in relation to one of your slides where you had the circles and the diamonds. Have the diamond terms been tested if they retrieve cases of Parkinson's?
Not Parkinson's specifically. It's a nice visualization. We've actually built a couple tools where you could interactively, you know, play with the semantic similarities. That's something I would like to also make available. So people who are interested potentially in reviewing SMQ codes, you know, I'm hopeful that this sparks new ideas beyond signal detection in different ways that semantic networks could be helpful in all kinds of data retrieval tasks.
Okay, great. Next question is, could this also be applied to coded data to identify signals from a safety database instead of a regulatory database?
I totally believe so. I will say our next steps are, you know, the initial experimentation was done in FAIRS, which is a very messy data source. I think our natural extension is to start looking at real world data, to look at things like clinicaltrials.gov. One of the pieces that I'm working on right now, I talked about this PV Lens reference database. We've currently mapped the PV Lens to the FAIRS publicly available data. We've roughly mapped somewhere between 96 to 98% of all FAIRS drug references to the PV Lens reference database. And what I'm working on as next steps are, for example, mapping clinicaltrials.gov drug references to PV Lens to make it sort of our bridge to perform these types of analysis across different data sets. I would say, you know, MedDRA terminology, I've been working with UMLS for 20 plus years. It's pretty, I'm not going to say it's easy, but it's much easier to deal with medical conditions than drugs. Drug references are notoriously messy, no matter what database you're looking at. And so that's where a lot of my current focus is.
Okay, thank you. Next question is somewhat related here. Can this method be used in clinical trials and not necessarily in a post-marketing pharmacovigilance safety signal detection setup?
Yeah, I think, you know, the sky's the limit potentially of how this could be utilized. Ultimately, what does the semantic network give you? It gives you a way, like we did early with, I mentioned this code slinger project. So if I was an epidemiologist interested in Parkinson's disease or myocardial infarction, you know, I still go and build a code list. no matter what data source I'm looking at. And I think building libraries of phenotypic data where you don't have to recreate these code lists is, where we would like to be, but we're not there yet. And I see the semantic networks as a bridge to helping people make these connections a little more quickly.
Okay, thank you. Next question is actually complimenting you on your presentation and is asking, without a lot of experience on Bayesian models, how do you begin implementing this type of methodology in, I guess, our signal detection process? So I guess they're asking, how would they get started doing something similar in their
Yes.
company?
So step one, you need the semantic similarity calculator, which I said we're working quickly to open source. I have developed both programming APIs. So the semantic server runs as a service. It's a Java-based application, but I know not all my statisticians can code in Java. So I've written both Python and R library bindings that allow them to connect, interrogate the semantic similarity server and say, tell me the distance across concept A and concept B under this representation and give me the scores back. And it does that in sub-millisecond time now. And so that's really the first tool set that you need. And then the second is that all of the Bayesian dynamic borrowing pipeline as we built it today exists in R code. So R is an open source statistical programming environment. And we're working on trying to at least get an outline of how you would perform the analyses to publish with the manuscript and put that on the GitHub as well. So I would say, if you're interested in this space, please stay in touch with us. We're happy to share what we can, and as soon as we get all the clearance, we'll be releasing the code as we can.
Okay. Next question is about the last slide where you referenced years. Are you referencing to time or to documented patient years? Or you may not have that in spontaneous reporting, so that would be interesting to learn.
In years. I'm not quite sure.
Referring to the last slide, apparently, in your presentation.
Yeah, so on this slide, we have the difference in time to detection. And so essentially what this is saying is these are the number of true positive signals. We define a true positive as meaning we know that the adverse event was added to the label within 1 to 3 years after the product came onto the market, right? And all we're saying here is that given the data that was reported in FAIRS, we were able to detect those events earlier than the traditional method. So it may not be that it was detected in years. I think what we really showed is that for newly launched products, the method was able to start to detect those signals by month 3 or 4 after the data was starting to show up, where the existing methods were taking much longer. That's not to say the existing methods wouldn't eventually have found them. It's just that we found them on average 5 months sooner than the traditional approach. I hope that clarifies.
Okay. Let me move on to the next question. What is your regulatory experience with using this method? Are there previous publications discussing its use in CTs? I guess that's clinical trials, but I'm not aware on regulatory position.
No, as I said, so Greg Powell on our team has worked with people at FDA for the last 25 years. He's had some sort of behind the scenes conversations with people there who have expressed that they're very positive that we're even developing new methodologies and that they have neither the time nor resource to do it on their own. And so I think any advancement in signal detection work is being seen as a positive. That's not to say it's going to be adopted tomorrow. We need much larger sample sizes than what we've shown, and we need to show over time systematically that it's going to perform at least as well, if not better, than existing methods. So it's a work in progress.
Okay. Next question is, how would you compare your new method with what's already in use for signal detection of signals? What is the success rate? What about the implementation of artificial intelligence? Will it go hand in hand with your method, or is it something totally different?
I'll answer the AI question first. I think there's a lot of interest in trying to get AI to do signal detection early on. One of my concerns, and I saw it when we were constructing this PV Lens database, and the reason I really went gung-ho on creating a natural language processing rules-based approach to creating this drug reference database is we wanted it to be fully replicable. We wanted to know if I ran it today, tomorrow, or the next day, I'm always going to get the same result back. And there are many projects going on across academic institutions of trying to use technologies like ChatGPT and LLMs to essentially extract the label information. What I was able to show and shared with some of those researchers is that the LLM technologies don't have a full understanding of measure terminology. It's just a fact. You can't expect ChatGPT to know all 26,000 measure terms because it's based on data that it's been trained on and seen in the real world. And in a highly regulated space like we live in drug safety, we need to know that, you know, all of the terms that are potentially there are fully represented. And so when I compared our data extraction to one of the more popular LLM projects going on, I found that they had missed over 1,500 measure concepts. And that's not to say that their methodology was wrong, it's just a limitation of the large language models and the AI tools that are not exposed to all of the measure terms. I think we could eventually train the AI tools to better understand and know how to use MedDRA or other biomedical terminologies, but they're just not there yet. And can you remind me what the first part of that question was?
Sorry about that. Let me see if I can get that one back.
And I'm a huge fan of AI. I'm just saying, when it comes to terms of knowing and understanding a language system like MedDRA, they're just not there yet, in my opinion.
Okay. So the first part of the question was, how do you compare your new method to what's already in use for detection of signals, and what's the success rate?
Again, we've only compared to the WHO's IC methodology. We compared it against traditional DPA methodologies. So I would say from our preliminary proof of concept experiment, it's showing strong promise, but there's no definitive answer yet.
Okay, let me move on to the next question, which is, I think, four more or so questions. So the next is interesting concept, but I hope I'm understanding the theory behind this. How do you determine the distance or measure of a PT terms? Is this more of statistical work or GSO? I'm not sure what that stands for, work.
It's purely statistical. If you're really interested in the nitty-gritty details, if you follow the links to the papers, we have all of the formula in their excruciating detail. As I mentioned, part of this semantic similarity calculator, we actually had to implement some of the metrics ourselves from scratch. They didn't exist in the Apache CTAKES project. And I had tested and verified that the values we were seeing from this code base matched exactly to what the old original Perl UMLS similarity package did, built by Dr. Peterson over 20 years ago. So that gave us some confidence that we were at least, we weren't fooling ourselves that we were making these computations much faster and getting different answers, but that we are actually seeing the same exact results.
All right, next question. In your example, would it support potential signals of worsening of Parkinson's analysis?
I'm not sure how to answer that question. I will say I spent 2 years working with the PPMI, the Parkinson's biomarker data set from the Michael J. Fox Foundation. A lot of that data is very, I'll say there are things like the MoCA score, so the Montreal Cognitive Assessment Test, not necessarily coded in terms of MedDRA. And so a lot of what I saw in working with that type of data is that you would measure, let's say, cognitive decline over time based on that assessment, but you wouldn't necessarily get anything from a semantic network that would help you there. It's more of a quantitative analysis. So I'm not really sure if we would see in observational data, different coding for different severities of Parkinson's over time. But it's an interesting question.
Okay. All right, next question is, based on your presentation, I got the idea that the method may be used to retest or test SMQs, possibly helping to create new ones. In other words, grouping terms related to an issue in questions.
Mm-hmm. Is that a question or a statement?
That's a statement. Sorry. Yeah. So it's a question followed by a statement.
Yeah,
grouping terms related to an issue.
I completely agree. I think, everyone wants to, even internally at our company, we don't have a policy of always following an SMQ. There are internal code sets that are built for condition XYZ. I think this is a tool that could help those clinicians who are in charge of building these code sets to potentially better explore. You can't know all 26,000 MedDRA PTs in your mind. And if you look at the entire MedDRA hierarchy, there's 110,000 plus terms in there. So any single human trying to understand and grasp all of that on their own is a huge burden. And I think these are tools that could definitely be developed to help improve the way that we do code building today.
Okay. All right, the last couple questions here. Can the semantic approach replace QMS or only complement them?
A QMS?
Yeah, let me ask them to restate. I wasn't sure the acronym myself.
Or was it an SMQ?
Maybe it's SMQ. Yeah, that could be possible.
Yeah, so I don't think it's going to replace them, but it could help the teams maybe reevaluate and improve them.
Okay. All right, next question is, you also mentioned that different representations of MedDRA might have an impact on calculating the distance. Can you elaborate on what kind of difference you were thinking of?
Yeah, so I don't think I mentioned which combination we actually settled on. So as I said, we looked at combining MedDRA with SNOMED, combining it with MESH, the medical subject headings, which is highly used on PubMed. And then we looked at combining all three. And what we found through our analysis was that MedDRA combined with SNOMED was the most performant. And I don't know if that indicates there's a limitation with the MESH terminology itself in the way that it maybe is taking away from the richness that we find when we just look at MedDRA with SNOMED alone. But we didn't look at any other terminologies because those are really the big three that cover medical conditions and adverse event relationships. And ICD-9 is old. ICD-10 is essentially a subset of SNOMED. So you can take with it that there wasn't really a need to test ICD-10 in combination. But I did find it interesting that MedDRA plus SNOMED was our best performant combination. And to me, that tells me there's potentially more testing to be done there. But I would say if you're going to take any steps working with these tools or technologies, I would just recommend going with MedDRA plus NoMed for now.
Okay, and the last question we have is it possible to see a small demo?
A small demo? Maybe. Let me see. Yeah, so this is the semantic similarity calculator. And this is not how my team interacts with it. This was basically built as a debugging tool to understand how the tool works. But we can look at, let's see, HLGTs. And we could say, so adrenal gland disorders. Actually, let me find the old classic. Okay, I do have a shortcut here. So we can search for the term nausea. We want to look at only PT codes. And so there's the MedDRA code for nausea. And let me, you know, it's always dangerous to do a demo in real life. Okay. And okay, the classic gastrointestinal. Let's see what we find. Okay, we'll just pick this, gastroplasty. So if I wanted to know the distance between these two terms, I've got gastroplasty and the PT for nausea, and I want to see what it looks like when I combine SNOMED with MedDRA. I can compute those metrics, and we see how quickly it returns. Here are all the scores against the Wu-Palmer, the LCH, the Resnick, and you know, it does that with relative ease. So if we wanted to then see how close is vomiting to nausea, my copy paste is not working so well. And again, for my programmers, I have an API where if they're writing in their R code, they can do this quite quickly. So we see for nausea and vomiting, it's no surprise that they're more related, and their Wu-Palmer score was 0.85. When we did nausea with the other concept, gastroplasty, it was like 0.33. And so this shows you how the distance computations would come into play. If you give me one more second. Okay. We can see the interactive. So are you able to see my screen now?
It's a little small.
Yeah, so this is showing essentially that Parkinson's disease concept, but extended even greater. And so I think I set the threshold much lower. So all the terms in yellow are being pulled in through semantic similarity. And again, we see over here, this hypertonia and muscle rigidity came from the narrow SMQ definition. But if we were to go from cogwheel rigidity, we can see dyskinesis, Morvan syndrome, pyratonia, benign congenital hypotonia. So this is what I was talking about when I said I'd like to see us explore building more interactive tools like this that would give you the ability to zoom in and really explore these semantic relations between the terms. So I thought it was pretty cool. I hope you can see the potential here of where we can go with this.
All right. Well, thank you very much, Jeffrey. That's all the questions that we have. So we'll turn things over to Amit to close out the meeting.
Great. Thank you so much.
Hello. Yeah. Well, thank you, Brian and Jeffrey, for the interactive question and answer session. And thank you, Jeffrey, for taking us through the interesting presentation and also sharing with us the history of semantic networks. It was good to see the valuable insights on the advancement that has been made in this area. I'm sure that this session has been very informative for all the people who have attended today. With this, I'll take on to the next slide for us. I'll quickly share my screen. Okay, as we come to the end of the session, what we want is to hear more from you. We'd like to have more information about what topics you would like to hear in the future webinar. If you are interested yourself to present any interesting matter related topic, we have the contact details on the slide right now. Please feel free to contact Martin or to the MSSO team and let them know if you are interested in presenting the topic. We would also like to remind you the next EU-MedDRA user group face-to-face meeting is on 22nd April 2026 in Hertfordshire, UK. And regarding today's webinar, please do provide us a feedback using the QR code that you can see on the screen now. We'll also send the link to the registered participants via email. Well, thank you everyone for today's session. It was wonderful. Thank you, Jeffrey, again, and thank you. Have a nice rest of the day. Take care. Thanks for joining.